Alright, hey everyone, so this is part 2 to the Operon video, and for this Operon I'm going to be talking about the tryptophan Operon. So, the tryptophan Operon is a repressible system, meaning that it is regulated by a repressor protein. It's quite different from the lac Operon which we spoke about in the last video, in which that is an inducible system and the presence of lactose will allow for the transcription of the genes. For the tryptophan Operon, the presence of tryptophan represses transcription. So, tryptophan Operon, repressible system, meaning that it's regulated by a repressor protein. I'm going to use purple. So, in conditions where tryptophan is present, the repressor protein would be bound to the operon. So, the entire point or purpose of this operon is to make the amino acid tryptophan. If you have tryptophan in the environment, it would not be energetically favorable to make more tryptophan since you already have some in the environment. So, in conditions when tryptophan is present, you will have the repressor protein binded to the operator sequence. Now, this operon has similar components to that as a lac operon, except the fact that they don't have the camp complex, and this has added regulation, such as the leader and attenuator sequence. So, if we look at this image here, we see we have the repressor gene, and similar to the lac operon, it's not a part of the actual operon. It is upstream from it, and this will produce a repressor protein. Now, the repressor protein has two sites, a site to bind to the operator, and another site for tryptophan to bind. Now, normally, what would happen when tryptophan is present in the environment is you would have the repressor protein here, and you would have tryptophan binded to the repressor protein. When tryptophan is binded to the repressor protein, that gives it the proper shape to bind to the operator. So when tryptophan is present, it's going to be binded to the repressor protein, and that's going to allow it to bind to the operator sequence. Now, the difference in the operon is, apart from the structural genes, the added layer of regulation in this operon, which is the leader and attenuator sequence. So the attenuator is a regulatory mechanism in the tryptophan operon that helps control the levels of that helps control if transcription occurs depending on the levels of tryptophan. And this is a secondary level of regulation. So if tryptophan levels are high, transcription for the mRNA structure will form, and that will result in the terminator conformation. So before I get into this, I kind of want to give a little, I guess, almost spiel on what the attenuator is. So attenuation, just start from the beginning because this is it'll make it a lot easier for you to understand how attenuation works and how the attenuator and the terminator, anti-terminator conformation occur and happen if you understand the conceptual aspects of it. So attenuation is essentially a mechanism for reducing the expression of the tryptophan operon when levels of tryptophan are high. Rather than blocking the initiation of transcription, which it cannot do, attenuation can prevent transcription from completing. So once transcription occurs in the leader sequence, right here, we begin to encode for the attenuator as well. Now, the attenuator is essentially just a smaller sequence located in the leader region, or the leader sequence. The attenuator does not code for any specific protein, but what it does code is for an mRNA strand. And the mRNA strand that it codes for has self-complementary sections that can form into various Harpin structures. It's also important to note that when thinking about this concept, you have to recall and remember that protein synthesis differs between prokaryotes and eukaryotes. Since prokaryotes, they don't have a nucleus, transcription and translation occurs simultaneously. So once RNA has began transcribing the once RNA has begun transcribing the operon, a ribosome is then going to attach to that newly made mRNA strand while RNA is still transcribing more of the mRNA strand. So when tryptophan is present, there's a large amount of what is known as tryptophan tRNA, and it's essentially just a tRNA charged with tryptophan. If there's plenty of tryptophan in the environment, then the ribosome will not have to wait long for the tryptophan carrying tRNAs and will rapidly transcribe the leader and attenuator sequence. If there's little tryptophan, the ribosome will stall and will have to wait until more tryptophan tRNA can be sent to it, allowing it to make more of the polypeptide structure. So, I guess why does that matter? Why does the rate of the transcription matter? So, if the ribosome translates slowly, it will pause due to the lack of the tryptophan tRNA, and its pausing causes the formation of what is known as the anti-terminator Harpin, which allows transcription to occur. So, the anti-terminator is essentially just a conformation that forms when tryptophan is low, and it allows transcription to occur. So it will allow transcription to continue. If tryptophan tRNA is readily available and there's a lot of it in the environment, then that will allow the ribosome to translate quickly. So that allows for the terminator conformation to form, making RNA. And so when the terminator conformation forms, that causes RNA to detach and end transcription. So it doesn't allow for more transcription to occur. So though it can start, let's say that there was a faulty repressor protein and it was unable to bind to it. In the case that there is high tryptophan in the environment, and this Harpin forms, RNA polymerase will be able to begin transcription, but once it gets to the Harpin, it's going to detach and it won't be able to continue transcribing the rest of the genes. So the terminator conformation is essentially just a conformation that forms when tryptophan is high and it doesn't allow for transcription to occur. So that is essentially all you need to know for the tryptophan operon, apart from mutations in the repressor protein. So R plus is going to signify a working repressor protein in which it has nearly the same structure as that in the lac operon. It'll have an area for the operator to bind. And it'll also have an area for tryptophan to bind. If you have a mutation, RB, that will result in a repressor that cannot bind to tryptophan. So the repressor instead will look something like this. It can bind to the operator sequence, but it cannot bind to tryptophan. And that will result in continuous expression. Since it cannot bind to tryptophan, binding to tryptophan is what allows it to bind to the operator sequence. If it's unable to bind to tryptophan, it will not have the correct shape to bind to the operator sequence, so RNA polymerase will be able to transcribe the rest of the genes. And there is a hierarchy you can follow. R plus, or the working repressor protein, is going to be higher on the hierarchy than RB, which is the mutated repressor protein. All right, going on to a practice problem. The table below lists genotypes of E. coli at loci required for regulation of tryptophan biosynthesis. The R gene encodes for the tryptophan repressor, the O gene refers to the operator sequence, the E gene encodes for the TrpE enzyme, the RB allele cannot bind to tryptophan, the E- allele does not produce any TrpE enzyme, and the OC allele cannot be bound by the repressor. Indicate whether you expect to observe presence of the TrpE enzyme when each genotype listed below is grown in a media supplemented with tryptophan and media lacking tryptophan. All right, remember, the Trp Operon, whole purpose is to produce tryptophan. So these enzymes that it's producing, the TrpE enzyme, is going to be a crucial enzyme in the production of the amino acid tryptophan. All right. So, assuming we have this genotype in which they're all working, and we have tryptophan in the environment, would we need to produce more of an enzyme to make more tryptophan? Remember, TrpE is going to make tryptophan. We would need to make more tryptophan. In this case, we wouldn't need to, since we already have it in the environment. It would not be energetically favorable, so TrpE would be absent in the media. Now, if we were to have a media without tryptophan, remember, this is trying to make more tryptophan. This enzyme right here helps code for tryptophan. For the making of it, we don't have any tryptophan in the environment, so we're going to want to make more tryptophan. So tryptophan will be present, or TrpE will be present in the environment. I would suggest that everyone tries to solve the rest of these problems, so you guys can pause the video, come back to it, or do whatever you want. All right, on to the second problem. We have a working repressor, a working operator. We have a non-functional TrpE enzyme. So we cannot produce any TrpE at all, regardless of if we have anything working. So we know that even if we have tryptophan in the environment, we cannot produce any TrpE. That's going to be absent. Without any tryptophan, we still cannot produce any TrpE, so it's going to be absent. All right, going on to this problem, we have RB, a mutated repressor, and everything else is functional. RB is going to result in constitutive expression, which I probably should have wrote that down here. Constitutive expression, since tryptophan cannot bind to it, R plus is going to result in pressable if there is tryptophan in the environment. So this will produce constitutive expression, meaning that expression is always going to occur. So we will have expression occurring if tryptophan is present in the environment and if there is no tryptophan in the environment. Going on to the other problem, we have all working genotype and we have a plasmid carrying a repressor protein. Remember, the repressor proteins are transacting elements, meaning that they can move from one plasmid or from one genotype to another genotype. This will produce constitutive expression. This right here will produce repressible expression in the presence of tryptophan. If we look at the hierarchy, we see that R plus is higher than RB. So this will take prevalence, or R plus will take prevalence over RB. So in media with tryptophan, if we have a working repressor protein, that will result in it being absent. In media without tryptophan, that will result in TRPE being present in the environment. For this bottom problem, we have a plasmid carrying an OC. So this OC will result in constitutive expression. However, this is a cis-acting element, meaning it's not movable. It cannot impact this genotype, not like the transacting element. So, if we were to have this working genotype, when tryptophan is present in the environment, we're not going to make any TRPE, so that's going to be absent. When tryptophan is not present in the environment, we're going to make TRPE, and so it's going to be present. This is how you solve this problem. However, if I'm not mistaken, I believe there is a mistake for this answer right here in the answer key. So when plugging it in, let me check what the answer says. Or I guess it was fixed. Never mind, the answer was fixed. This is still the correct answer. So this is the answer to one of your practice problems. Hope you enjoyed this video. Yeah.